Membrane receptors are crucial for the human body. They allow our cells to communicate with the outside world. Looking at a cell, the phospholipid bilayer, which is semi-permeable, has a membrane receptor that is responsible for communicating with the outside environment. Each specific receptor only binds with specific signaling molecules, or ligands. This is why only select things enter the cell. Signal transduction is where a ligand binds to a membrane receptor and causes an intracellular response. There are hundreds of different membrane receptors and ligands. But now, looking at a specific membrane receptor, the G protein coupled receptors, or 7 transmembrane alpha helices, we can see how G proteins and salicylic AMP interact. In this example, our ligand will be adrenaline. When adrenaline binds to the receptor, a conformational change occurs and activates the G proteins, gamma, beta, and alpha. The GDP, which is guanosine diphosphate, di meaning 2 phosphates, is attached to the alpha subunit. During this conformational change, GDP is exchanged for GTP, which is guanosine triphosphate, tri meaning 3 phosphates, which are located in the cytosol of the cell. The alpha subunit and the GTP then dissociate and bind to the effector protein, which in this case will be adenylated synthase. This protein then converts ATP to CAMP and releases phosphates. CAMP is the protein's second messenger, which can cause a variety of effects. In our case, increased heart rate, which makes sense since our ligand was adrenaline. CAMP eventually does convert back to ATP. After this, GTP is hydrolyzed back to GDP. The alpha subunit and GDP then reattach to the beta and gamma subunit, and thus completes the G protein signaling process.